Thank you.
Okay.
Good morning, dear colleagues. Thank you for joining my lectures. Its title is Drug Design and Repurposing, an overview with selected examples. First of all, I would like to express my gratitude to organizers for the opportunity to share some general ideas and practical experience in one of the most rapidly evolving areas of natural science, drug design. Uh, Today I will talk about scientific field called drug design. Drug design is a great example of scientific integration. It brings together medicine, chemistry, pharmacology, biotechnology, molecular biology and biophysics, bioinformatics, computer modeling, and even AI technologies and quantum computer. At the same time, it is an applied discipline within the natural science, and it plays a key role in both molecular pharmacology and modern medicine. Drug design has reached its current level only recently. However, drug design is still a slow and very expensive process. It can make many years and billions of dollars to bring a single drug to market and to patient. Today we can cover only a small part in this lecture, but I hope to give you an introduction to this fascinating area of modern research. The plan we can see on the slide. We'll start with some basic concepts in drug design. Then I briefly touch on history of the drug development. After that, we'll look at few examples of rational drug design and drug repurposing. Finally, I'll share a few concluding remarks. Since drug design involves many online tools and resources, I have also included some supplementary materials with useful links and information, so you can try out some of these tools yourself. And now let's get started. What is drug discovery or drug design as a process? Often the term drug discovery and drug design are used interchangeably, but drug discovery and drug design are not the same. On the left side of the slide, you can see a diagram of the drug discovery process. It shows the main steps involved. The process starts with an idea to develop a specific drug. That is early drug discovery or basic research. At this stage, we try to understand the molecular mechanism of the disease. Then we identify molecular targets, the key molecules involved in this disease. These targets help us find potential drug. Next, we begin searching for drug candidates using structural, chemical, and pharmacological databases. At this point, we have a huge number of compounds. Our task is to narrow them down. We use in silico and in vitro methods to select the most promising ones. The selected compounds become drug candidates. It is the result of drug identification stage. Then drug candidates go through the in vivo test during preclinical research and next go through stage of clinical trials. In the end, only one drug may show good therapeutic effects and be approved to medical use. So, drug discovery is a broad process of finding new potential drugs. Drug design is more specific part of drug development. It focuses on creating molecules that interact well with a biological target, for example, with a protein involved in disease. So, in short, drug discovery is the full process of finding and developing new drugs, from basic research to clinical trials and drug approval. It uses computer tools, lab experiments, and clinical research, or in silico, in vitro, and in vivo studies. Drug design is focused on the early steps from 1 to 3, as we can say on the slide on the right side, and usually does not include animal and human testing. It is limited in silico and in vitro studies. And today we will focus on this part of drug discovery. The drug discovery includes several stages, many of which involved in silico methods. These are computer-based techniques used for screening of database, calculating various molecular and pharmacological properties. There are also methods for investigating complexes between molecular targets and potential drugs by computer modeling. All these approaches are integrated in the rational drug design. So let's briefly go over the main steps of rational drug design. You can follow them on the slide. The first is target identification and validation, or finding a biological molecule involved in the disease. For this, you need to understand the cause of the disease at the molecular level. For target identification, researchers use 3D structural information obtained by X-ray crystallography, cryo-electron microscopy, or nuclear magnetic resonance spectroscopy. Then we need to select from the databases a set of compounds that may fit the active site of the target molecule, like a key for a lock. This is the lead discovery and optimization stage. Then, thus, rational drug design focus on the early stage of drug discovery. It involves usually in silico methods to predict interaction between molecules, but also the in vitro methods. And now let's take a look at the stage of lead discovery and optimization in rational drug design. It quite complexes and included several procedures. You can see the schemes and results on the slide. The first step is hit finding and validation. Here we need to define what is hit and lead compounds. Hits are compounds with some activity against their molecular target. Lead compounds are improved versions of hits that serve as a foundation of future drugs. These compounds aren't yet a drug, but they are potential starting points. During the hit finding and validation, we try to find compounds that bind to the target of our interest. If we know a structural element that can act as a key fragment of the future drug candidate, we can place this fragment in the possible binding site of the target molecule. For this, we use special software, usually integrated in the special databases. Then we add different chemical groups to fill the area of binding site. This procedure involves screening small chemical fragments. Key criteria for such fragments are the following: low molecular weight, usually less than 250 Dalton, high solubility and chemical stability, low complexity or low number of rotatable bonds in the ring system, no more than 3 hydrogen bond donors and acceptors. Finally, a ligand is constructed fragment by fragment in the active site of the target. We also must consider the charges distribution, the polar, hydrophilic, or hydrophobic features of the target binding site. As a result, we design the ligand which in general fits to the binding site. It is the algorithms of fragment-based drug discovery. The next is hit to lead stage. This step involves modifying and selecting a number of hit compounds and evaluating their potential to become actual drug candidates. After that, we move to lead discovery and optimization, where lead compounds are further modified. Key tasks of these tasks are maximizing potentiality, selectivity, and improving their pharmacokinetics. Here, potential shows how strongly a compound interacted with possible targets. Selectivity shows how various compounds act on one target. Pharmacokinetics show how the body absorbed, distribute, metabolize the compound. These characteristics are available in the database for known compounds and can be calculated for new ones. So, in conclude, the main goal is to develop a drug candidate for preclinical and clinical testing. So we can see hit as a huge number of compounds. Then we limited it for their specific abilities to interact with targets, and they have some properties which are very important for their using in the clinical in future for clinical trials, not even as a future drugs, as I said. And this is a so-called absorbance, distribution, metabolism, extraction, and toxicity. This set of parameters we can calculate in different types of databases for special tools integrated in such databases. Then look at some methods in rational drug design. First of all, is virtual screening is a computational technique to identify potential drug from large chemical libraries. It involves the evaluation a lot of small molecules to predict their ability to bind to a biological target associated with a particular disease. For this, one can use computer-based or in silico methods. There are some types of virtual screening. Pharmacophore-based virtual screening starts by identifying the essential chemical features required for molecule to interact with its biological targets. This makes up the pharmacophore. Structural-based virtual screening requires the 3D structure of target molecule. It often involves molecular docking, where candidate compounds are virtually fitted to enter the active site of the target to estimate binding affinity and interaction patterns. Ligand-based virtual screening, in contrast, is used when the structure of target is unknown, but information about known active compounds is available. It bases on molecular similarity to identify compounds with similar properties. So virtual screening allows research to significantly reduce the number of compounds that need to be tested experimentally, thereby saving time, costs, and resources in the drug development process. So molecular docking is one of the very interesting components of virtual screening. Molecular docking is a method that enables the prediction of how existing drugs might bind to a new target by virtual screening chemical pharmacological databases. What steps are involved in molecular docking? First, we obtain 3D structure of biological target from structural database, such as, for example, protein data bank. Next, we select an appropriate ligand library, and virtual screening is performed using molecular docking. Here, we estimated the binding energy between two molecules as they form a complex. The docking procedure involves generating multiple possible position and orientation of ligand at potential binding sites on the target molecule. For each of these configuration, the binding energy is calculated. The macromolecule is surrounded by a three-dimension grids. The distance between two adjacent grid points are near the 1/4 of length of single carbon-carbon bond. Each atom of ligand atom is placed in each grid point, and the atom-atom interaction energy is assigned to this point. So the goal is to identify the structure with the lowest that it is most favorable free bending energy, named docking score. This structure represents the most likely configuration of the complex from an energetic point of view. How we can evaluation the binding energy between ligand and molecular target within molecular docking approximation. To do this, we use special scoring functions. A typical scoring function, as shown on the slide, is a weighted sum of different energy components: van der Waals interaction, hydrogen bonding, electrostatic interaction, torsional or rotation entropy loss, desolvation or dehydration energy. The first three terms describe intermolecular interaction in a vacuum. Van der Waals energy is calculated using the Lennard-Jones potential. Electrostatic interactions are described by a screened column potential. A distance-dependent electric function here mimics solvent screening. Hydrogen bond energy is calculated using 10-12 potential with some empirical parameters. Torsional energy represented the loss of rotational degrees of freedom in the ligand upon binding. It's proportional to the number of rotatable bonds formed by heavy atoms in the ligand. Desolvation energy represented hydrophobic hydrophobic interactions caused by water displacement during binding and estimated how much of the ligand surface is buried by the macromolecule. All potentials depends on atom types in molecules according to the force field concepts. Thus, the docking method is an effective approach for screening a large number of ligands to evaluate their interactions with target molecule. Molecular docking significantly accelerates the identification of potential drugs. Thousands of compounds can be screened within just a few hours. This greatly reduces the experimental investigation. And one more tools for the which is used in rational drug design. It's a high throughput screening or an experimental method that involves testing of real chemical compounds in the laboratory. The goal is to identify active compounds through molecular and cell-based samples or assays. High-throughput screening is often combined with biophysical experimental technologies such as mass spectrometry, colorimetry, and others, as well as bioinformatics. Such approach provides the understanding of compound activity and mechanism of their action. The key difference between virtual and high-throughput screening is that virtual screening is a computation or in silico process, while high throughput screening is a physical or in vitro process. Virtual screening is faster and more cost-effective, but it depends on the quality of computation models. On the other hand, high-throughput screening provides experimental data, so it's more expensive and time-consuming. So we talk about some general concept of drug design. And now I want to share with you some example of drug design. Maybe it's was valuable for you because we from abstract describing can show the real experiment and the real process. My interest in developing new therapeutic agents began when we took part in international research project. You can see it title on the slide. The project lasted for 3 years and involved several research groups from United Kingdom, Germany, Mexico, Russia, and Ukraine. The main goal of the project was to create an analog of anti-cancer drug design actinamide CND, or IMD in short. IMD is an anti-cancer drug used to treat various types of tumors. It works by binding to DNA, which prevents cancer cells from growing and dividing. However, it causes side effects such as liver and skin issues, muscles pain, and hair loss. That's why one needs to develop alternative compounds that can bind more strongly and selectivity to DNA. What is IMD from them? For their chemical structure. It consists of an oxazone chromophore with two identical side chains. Its anti-cancer activity is based on the intercalation of the planar chromophore into GT-rich DNA sequences. The side chains lie in minor groove of the DNA and additionally stabilize DNA ligand complexes. This blocks protein-DNA interactions, stopping DNA replication and preventing cancer cell proliferation. So now we knew both the possible cause of diseases and the molecular target, or DNA. The next step was to design new molecules with anti-cancer activity similar to IMD. These molecules need to include the phenoxazone chromophore, which is essential for DNA binding, as well as appropriated side chains. By comparing the anti-tumor activities of compound with different terminal groups in side chains, such as amino, dimethylamino, and diethylamino, we have chosen how to modify the side chains. These new compounds were a potential hit candidates. Then we synthesized these compounds and tested their anti-cancer activity using cancer cell cultures. From this test, we identified 4 promising ligands, each containing between 2 and 5 methylene groups in the side chain. We named them Act 2 to Act 5. This step corresponded to the heat validation stage and is similar to high throughput screening or in vitro functional assays. Next, we wanted to understand how these compounds interact with DNA. For this, we used several biophysical methods. Ultraviolet visible spectrophotometry helped us examine different modes of ligands binding and also indirectly estimate their stability using a constant of binding constant. Different scanning calorimetry gave us information about the thermal stability of DNA ligand complexes. Nuclear magnetic resonance provide detailed structure features in the binding site. This method is part of binding assays with high throughput screening. They measure how strongly various drugs candidates bind to their target. Stronger binding generally means greater potential effectiveness. Based on these studies, we found that a compound with 2 methylene groups in side chains, or ACT2, showed the strongest interaction with DNA. So we selected it as our lead compound, and this action corresponded to lead identification stage of rational drug design. We also applied computational methods to support our experimental data. This includes molecular docking, Monte Carlo, and molecular dynamics simulation. This simulation confirmed the DNA ACT complex was stable over the time of simulation. They also showed that their stability is due to additional hydrogen bonds in the and specific interactions with water molecules at the binding site of the target. In this way, these computational tools served as a kind of virtual screening. In conclusion, our study showed that experimental and computational combined approach can be used to determine physical parameters of drug-DNA interaction. These parameters help us understand and predict a drug's activity in vitro. At the end of the project, we were able to make a recommendation for possible preclinical trials based on our findings. So we move from our first part of the lecture, and now after our brainstorm session, let's take a bit relax. I'd like to share with you some interesting facts from the history of drug design. How did people search for medicine long before modern science and pharmaceutical? This was a long period from ancient times up to 19th century. The so-called algorithm for find medicine was very at the time was very different from what we know today. Nevertheless, people had a system. The main method for discovering drugs was observation, trial, and error. People carefully observed nature and tried to understand what caused disease. They experimented with natural materials to see if this could help. Natural materials were collected and prepared in various forms, as teas, various extracts, and salves for external use. Some drugs have known since from these times. For example, morphine, derived from the opium poppy, is an analgesic and psychoactive drugs. Quinine was the first effective treatment for malaria. It was obtained from the bark of Radiophysics tree. Salicin was the first natural substance used for pain relief and fever reduction. It found in the bark of the willow trees. People also began to create early version of modern drug database based on their knowledges. The example of this first drug database was the following. The Ebers papyrus from ancient Egypt, dating from around 1,500 years before Christ. It contains 877 prescriptions for various diseases. Other example, it is Scorda's Materia Medica, written during Roman times, which describes treatments based on plant, animal, and mineral products. Synthetic drugs produced by chemical methods began to appear in the 19th century. Morphine was first isolated from opium, but not synthesized as a chemical compound it was isolated from opium in 1804 in Germany. In 1827, the German pharmaceutical company began the commercial production of morphine. Quinine was first isolated in 1820 in France and salicine in 1828 in Germany. Later, in 1897, a German chemist, Felix Hoffmann, from Bayer pharmaceutical company, synthesized a more stable form or acetylsalicylic acids. This new drug was named aspirin. It became one of the first modern medicine, and it's still widely used in modern medicine. During the 19th century, scientists started to really systematize knowledge about drugs. One of the first manual databases were pharmacopoeias and chemical references books. A key example is the United States Pharmacopoeia. It collects and describes medicinal substances, their preparation, and quality standards. In Europe, similar works included the British Pharmacopoeia and Pharmacopoeia Londinensis. Chemical encyclopedias like Gmelin Handbook of Inorganic Chemistry began publication in Germany. So, although these early databases were printed books, they served the same purpose as modern databases, to collect knowledge about drugs for professional use in medicine and science. They were like manual databases for doctors and pharmacists. The most famous event in drug discovery at early 20th century was the discovery and development of antibiotics. It began in 1928 when Alexander Fleming, a Scottish bacteriologist, accidentally discovered penicillin. Fleming noticed that a mold they identify as a penicillium notatum had killed bacteria in one of his petri dishes. Then he extracted the substance produced by pure mold culture. This substance inhibit bacterial growth. It was named penicillin. This accidental discovery became one of the most important medical achievements in history. That is why the mid-20th century is known as the golden age of antibiotics. Fleming was awarded the Nobel Prize in Physiology or Medicine in 1945. The chemical structure and formula of penicillin were first determined by Dorothy Crawford Hodgkin in 1945. She used X-ray crystallography and by analyzing the diffraction patterns, solved the structure and built a physical model of penicillin. As a result, she revealed the beta-lactam ring, which is crucial for penicillin's antibiotic properties. For this work, she used Alan Turing computer. Hodgkin was awarded the Nobel Prize in Chemistry in 1964. The discovery and development of first antibiotics followed several key steps: observation, isolation, mass production, purification, and clinical use. So the concept of drug design aren't born at that time. The concept of systematic drug design is only about 50 years old. In 1988, James Blake, Gertrude Elion, and George Hitchens were awarded the Nobel Prize in Physiology and Medicine for their outstanding work in this area. Their research led to development of new drugs that became essential for treating a wide range of diseases. However, their research had a more fundamental significance. Before this, drug discovery mostly involved chemical modifying of natural substances. Blake, Elion, and Hitchens introduced a more rational and scientific approach. So, what exactly did James Blake do? He designed a drug called propranolol and described its molecular mechanism of action. Propranolol was the first antagonist of beta-adrenergic receptor. By studying how its drug interacts with their receptor, James Black discovered that propranolol binds to the beta receptor. As a result, natural substances of this receptor, adrenaline and noradrenaline, can no longer trigger the usual fight or flight response. This leads to reduction in heart rate and blood pressure. Taking propranolol effective for treating conditions like heart attacks and hypertension. Today, we can say that the discovery of such drug is a good example of ligand-based virtual screening. Since the receptor structure was unknown, the first 3D structure was received only in beginning of 21st century. One can use known active ligands to build pharmacophore models and applied searches and analysis of physicochemical and biomedical properties to identify new hit compounds as propranolol finding by James Black. And what about Gertrude Elion and George Hitchkins? They won the Nobel Prize for development at a completely new way of designing drugs by rational scientific approach. One of their key insights was that cancer cells divide very quickly and needed a constant supply of DNA and RNA. To stop this process, Elion and Hitchkins developed drugs as analogs of DNA and RNA building blocks. They used purine and pyrimidine derivatives to block enzymes essential for DNA replication and RNA processing. Their research led to development of many important drugs. Some of them are represented in slide. This drug contain to save lives today also. So we can say the discovery of these novel anti-cancer and antiviral drugs is also a good example of ligand-based virtual screening, because the detailed structure of some essential molecules for DNA replication, such as enzymes shown on the slide, was then unknown. And now we also can describe after just going over the basic definition and main stage of the drug discovery, a close look at how new drugs are actually developed. We'll do that using a classic example, the development of drugs to treat acquired immune deficiency syndrome, or AIDS. This is one of the earliest and most well-known cases of drug creating using rational drug design. As we mentioned earlier, the first step of drug design is to understand the disease. AIDS is caused by a virus known as a human immunodeficient virus, or HIV. This virus infects T cells or T lymphocytes that plays a crucial role in the human immune system. So first, researchers need to study the life cycle and structure of HIV. This helped them identify the molecules essential for virus replication. These molecules became the molecular targets for antiviral drugs because potential antiviral drugs should block or inhibit them. What is HIV? HIV is a retrovirus or RNA virus within a lipid envelope. Inside the HIV contains 2 copies of single-stranded RNA. HIV converts its RNA genome into DNA, which then integrated into the host genome. The HIV genome encodes 8 viral proteins, including reverse transcriptase, integrase, and protease, which are essential for virus to complete their life cycle. So, what is its life size? HIV target T cells in immune system. First, HIV binds to the surface of T cells using cell receptors, CAD4, and 2 co-receptors. Then HIV releases its RNA into the cell. The enzymes reverse transcriptase convert the RNA into DNA, which then integrated in the host genome by another enzyme integrase. Then the virus must first mature, and it uses HIV proteases to do that, cutting viral polyproteins into functional parts. Then the infected cell starts to produce new viral RNA and proteins. These components are assembled into new virus particles, which eventually leave the cells for infecting others. Drugs used to treat HIV were developed based on the virus life cycle. As we mentioned earlier, several crucial proteins involved in life cycle and have been identified. Today we will focus on one of them, the HIV main protease. The 3D structure of this protease was successfully determined in 1989 using X-ray crystallography. This discovery accelerated the rational design of anti-HIV drugs. Structure of protease. Protease is a small homodimeric enzyme. It consists of two identical protein chains, each made of 99 amino acids. The homomer can exist in both open and closed conformation, which are essential for substrate binding and release. At the interface between two chains lies the active site where the enzyme cleaves viral proteins. This site contains so-called catalytic triads of amino acids asparagine, threonine, and glycine. This motif is typical for aspartatic proteases. The enzyme has also flexible flaps that cover the active site. The flaps can move up to 7 angstroms when binding a substrate. Main job of HIV protease is to cleave the large viral polyproteins. The cleavage occurs at the active site where two aspartate residues play a key role in the catalytic activity. Therefore, to block HIV replication, we need to design drugs that can bind tightly to active site of protease and inhibit its function. At the time, several proteases inhibited for other aspartate proteases, not for HIV protease, were already known. This helped researchers understand how to design molecules that could block specific protease, HIV protease activity. One of them, bestatin, is shown on the slide. However, after lab experiments, these compounds were not specific, were not specific to HIV. Drug development based on X-ray crystallography of HIV protease structure and was an example of structure-based drug design. The early version of the modern database have been already used in that time. These resources support early drug design along with manual screening and experimental virology. A key results came in the later of 1980s and early 1990s, when several HIV protease inhibitors were discovered and tested. Let's take indinavir as an example. The illustration shows that enzyme's active site at the center of the lies on the center of substrate tunnel. Idinavir binds tightly across the active center of the protease, making close contact with residues of both monomers. This binding induces a conformational changes. As a result, the flexible flaps close that block the enzyme's activity. You can see also some example of different HIV protease inhibitors, such as ritonavir, idinavir, and nelfinavir. And you can notice that their structure are quite similar. These drugs mimic natural protein chains, which allows them to bind to the enzyme like natural substrates. However, they are much more chemically stable than real proteins. Therefore, HIV protease can't cleave them, so they stay bound in the active site and block the normal function of enzymes. The development of this inhibitor is a classical example of structure-based virtual screening. Scientists use the crystal structure of protease to screen large libraries of compounds using also a molecular docking. They predict how well each compound would bind to the active site. In conclusion, based on this example, I would like to note that the following point. Researchers were able to discover drugs uh, uh, that inhibit specific molecular targets by studying the causes of diseases and identify the proteins essential for their development. The identification of the protein targets and their inhibitors involve a combination of medical, pharmaceutical data analysis, molecular and structural biology, database screening, and computer modeling. And now let's move to the next topic, or drug repurposing. What is drug repurposing as a process? Drug repurposing means finding new medical uses for existing drugs. These may be drugs that are already approved or still experimental. This approach is not new, but over the past decade, it has acquired significant attention. Today, about 1/3 of all drug approvals come from repurposing. Why is drug repurposing is so important? It is often faster, cheaper, and safer than developing a new drug from scratch. That's because we already know a lot about the drug's mechanism of action and how it behaves in body. But research, whatever, need to add identification biological mechanism of diseases and validation the new molecular targets. Researchers use several methods to identify new use for existing drugs. This includes searching through the medical literature to understand the nature of diseases, to identify and validate targets. Then testing large libraries of compounds to identify candidate drug. Techniques including computer modeling for virtual screening and various experimental methods for high-throughput screening. This could be done through lab researches in vitro or computer models in silico. Next, in vivo methods are used. The candidate drug is tested in preclinical clinical trials to ensure its safety and effectiveness for new condition. Finally, regular approval is obtained for new indication to explore the drug potential for a new purpose. In summary, drug repurposing is a promising strategy that helps speed up the discovery of treatments and reduce development costs. Now, let's look for examples of drug repurposing. The most recent examples of successful drug repurposing is the search of treatments against coronavirus, the cause of COVID-19. At the beginning of the pandemic, a wide range of antiviral drugs was already known. For example, drug development for treat HIV had already been through the study. In addition, many compounds have been created for treatment of hepatitis C and the Ebola virus. It is also worth mentioned that a similar attempt to repurpose existing drugs was made earlier during the first coronavirus epidemic. Let's take a look at the step involved in the repurposing process. The first step is understanding the nature of the disease of COVID-19. The structure of coronavirus includes main structural proteins. Spike glycoprotein or S protein is crucial for entry into human cells. Membrane glycoproteins and envelope proteins both contribute to virus outer structure. Nucleocapsid proteins binds to the viral RNA to form a protective structure around the genome. The genetic materials of coronavirus is a single-stranded DNA. This RNA is wrapped and protected by nucleocapsid protein. Together, they form the nucleocapsid. Membrane glycoprotein and envelope protein are involved in the packaging of nucleocapsid for virus integration. If we can see the life cycle of coronavirus, it has been studied in great detail. It begins when spike protein on the surface of virus recognized and bind to a receptor of human cell as angiotensin converting enzyme, or ACA2. This binding causes the fusion of the viral envelope into the host cell membrane, allowing the virus enter the cell. After entry, the viral genome material is released in the host cell and translated by host ribosome. The translation production, two large polyproteins are cleaved into 16 non-structural proteins by two viral proteases. So, as we know now, antiviral therapy is designed to disrupt different stages of coronavirus infection cell. In the virus, it's enabled to produce the essential components it can't replace or spread through the body. One of the most important targets is the spike protein. This protein allows the virus to recognize and bind to the host cell receptors, facilitates the fusion of viral and host membranes, and enable the viral RNA to enter the cell. Spike protein is a trimer. Each identical monomer consists of two subunits, S1 and S3. The S1 subunit is responsible for attaching the virus to the host cell by binding to the cell receptor on the surface of human cells. It contains three key regions, especially receptor binding domain and receptor binding motif, is a major focus for antiviral drug development. The SIDV subunit is involved in membrane fusion. The second key proteins are the main protease and papain-like protease. They cleave a large viral polyproteins into smaller functional units. That is essential for forming the viral replication complex. By focusing on these essential viral components, antiviral therapies can significantly reduce the virus ability to replication, spread, and cause diseases. What is the structure in the 3D structure of main protease? The main protease of coronavirus also plays a crucial role in viral replication by cleaving the viral polyproteins in specific site. It is a homodimer. Each monomer consists of three domains. Domain 1 and domain 2 contains the substrate binding site. Domain 3 is involved in dimerization. Dimerization is essential for enzymatic activity. The active site lies in the cleft between domains 1 and domains 2 and contains a catalytic diet composed of cysteine and histidine. This diet is crucial for protease's ability to cleave polyprotein substrates, which is essential for viral replication. The substrate binding site, where the substrate interacts with the enzyme, is located between two residues. Such 3D structure of main proteases as a key target is essential for antiviral drug design. Let's look at the process how the inhibitors of main protease were designed. Several known drugs have been repurposed as a potential inhibitor of coronavirus main protease using 3D structured main protease and molecular modeling as a part of virtual screening. We already know the general step involved in the drug repurposing process known main protease inhibitors. First, we need to analyze the structure of main protease, especially the active site. Then we screen existing inhibitors or other compounds to identify potential hits and lead compounds based on the molecular docking and molecular dynamic simulation. The resulting protein ligand complex are evaluated by their binding energy to estimate their stability. Another useful parameter is the root mean square deviation. This value measures the average distance between atoms in the original X-ray structure and predicted docking structure. A low root mean square deviation generally indicates a more reliable docking results. On the slide, we can see an example of complex formed by between the main protease and drug known as a methyl acceptor inhibitor. Its binding to the target was predicted using molecular docking as a part of a virtual screening. The structure of this inhibitor resembles a fragment of peptide chains, uh, similar to natural substrates of protease. In addition, it includes chemical groups that allow the ligand to form the energetically favorable complex according to the docking scores. So it was one of the great examples how the inhibitors of HIV protease can use for new clinical using. One more example, drug repurposing with the aim, it's the drug repurposing with aim to block S protein. In this case, we focused on the blocking the binding of 3 coronavirus to the host cell receptor. The aim of the study was to identify a potential inhibitor of the spike glycoprotein and could bind at the receptor binding domain interface with cell receptor. To achieve this, we began by using molecular docking to screen a list of 327 compounds from one of pharmaceutic database. This list included molecules with known antiviral activity, among them eye-approved drugs and natural products used in traditional medicine. One of the list is amantoflovin, a biflavonoid found in many plants, including, for example, ginkgo biloba. It has antioxidant, antiviral, and anti-inflammatory properties, and it is widely used in traditional medicine. All compounds in this database are commercially available. For the docking study, we used the structure of receptor binding domain binding with host receptor obtained from protein data bank. Docking was performed using Autodock Vina software. We started with targeted docking, focusing on the region of direct contact between RBD or receptor binding domain binding and cell receptor. Next, the 20 top ligands ranked by predicted binding affinity from molecular docking were subjected to blind docking. This step helped us verify whether the cell receptor interface is preferred binding site on the receptor binding domain for these compounds. On the slide, residues of RBD interacting with one of the investigated inhibitor or anti-hepatitis C drugs ledipasvir are shown. So we can see that some tools of molecular docking help you to understand the interaction ligand in the binding site with the target in detail and determine the type of interaction which stabilized them. So we can build the complex with our molecular target and investigated drugs. And we, interestingly, that was a particular interesting that the anti-hepatitis drugs and natural products or amantoflovin bind to different sites on the RBD. So then can block the different part of the site where receptor binding domain of coronavirus and the receptor of host cells can make a complex. So it's was an idea to provide to fulfill the computer modeling with a ternary complex when we combined two possible inhibitors on such complexes. For this, we do the molecular dynamic simulation. And so while dogs give us a quick estimation, molecular dynamics provides a valuable post-processing step to improve accuracy and confidence of the results. The molecular dynamics trajectories of ligand-target complex were analyzed to estimate their stability based on binding free energy. The molecular dynamics trajectory of the complex can be used to estimate this energy. Common method is molecular mechanics generalized bond surface area. On the slide, we can see this approximation for calculation of binding energy on the slide. The first three terms in this formula are standard to molecular mechanics energy terms, electrostatic and van der Waals interaction. The salvation-free energy includes the polar and non-polar contribution. The last term is the loss of entropy estimated by a normal mode analysis of vibrational frequencies. It's important to note that the accuracy of this simulation depends on specific parameters. The force field charges model, a model's treatment of interaction, entropy calculation, and so on. For this reason, the results are not always directly comparable to experiment values. At the same time, the relative affinity of ligands to the same target are useful, well reproduced. So, based on the result of these results of computer modeling or virtual screening, we can conclude that two anti-hepatitis C drugs in combination with natural biflavonoid amantoflovine are the promising candidates for blocking the receptor binding site on the receptor binding domain of spike protein. This serves as an example of repurposing existing drug for potential new application against coronavirus. So, at the end, I can make some final remarks. First, we can evaluation of drug development in a history period. When we look at the history of drug discovery, we can divide it into several major periods. Each stage reflects how progress in science and technology has directed scientists in discovering new medicine. The earliest period of pre-modern era lasted from ancient times up to the early 20th century. During this time, the discovery of new drugs were based on observation, trial, and error. The first phase was determined by luck and chance. Nevertheless, some important drugs were discovered during this time, including aspirin and penicillin. The second phase is characterized by the development of large chemical libraries, which could be tested in vivo and involved into the discovery of thousands of new drugs. This period led to a revolution in medicine. In 21st century, we entered in third phase. With a complete of human genome and valuable genetic information, drug discovery became more precise. In the fourth phase, it became possible to develop targeted specific drugs for individual patients. Finally, in ideal based on all these achievements mentioned above, we are entering in the fifth phase, also known as preventive, personalized, and predictive medicine. And a new term ad hoc drug means drug for the purpose only. Such drugs are often created to repurpose to treat an individual patient or a disease, but is a little ideal schemes. Also, we can remember that there are some stage of modern drug discovery. Now we know the main steps of it. It starts with an idea, developing a specific drug. We try to understand the biological and molecular mechanism of diseases. Then we identify molecular targets, the key molecules involved in the diseases. These targets help us find potential drugs. Next, we search for active compounds in chemical and pharmacological database. The selected compounds become drug candidates. These candidates then go through preclinical testing, including in vivo studies. In the end, only one drug may show good therapeutic effects and be approved for medical use. So, drug discovery is a complex process of finding new potential medicine. And the end, a final remark 3, traditional drug discovery versus drug repurposing. Traditional drug discovery is a long, expensive, and high-risk process that starts from scratch. This entire process can take over 10 to 15 years and cost billion of dollars. In contrast, drug repurposing focusing on finding new therapeutic use for existing drugs. Since the pharmacological and clinical profiles of these drugs are already known, the process is faster, cheaper, and less risky. In summary, while traditional drug discovery develops new drugs from the ground up, drug repurposing give a new life for existing compounds. There are one example for comparison the drug discovery from the scratch and it repurposing for new therapeutic use and the formula of these ligands. So I finish my lecture and my tip for the young for the students and young researchers is the follow. Working with database and using in silico methods for rational drug design often involves trials and errors. This is how you gain the experience necessary for success. And for my experience as a supervisor, I can say that many students learn and use these methods for some months during they fulfill their diploma thesis. So you can reach your success if you want to do it. Thanks.
Thank you, Anna, for this excellent lecture and very detailed on the drug design and drug repurposing. Now we have a time for questions. First, maybe we can start from the audience in Kiev. Are there any questions?
Yes, can I ask question? May I ask question?
Yes, of course. So please.
Thank you. Thank you very much, Anna Viktorovna. Very nice presentation and very informative and many interesting details were in this lecture. And as I understand, one of the key methods that you use in drug design is molecular docking. And this method is very powerful and very resultative. So we can use it for to obtain some information which can help us to understand how the molecule bind to protein or DNA. And I would like to understand just for myself, when we use the scoring function, so this is something like potential functions which we use in our molecular modeling, we try to approximate different contributions to the energy, to the interaction energy. And after that, we always want, of course, this is natural, we always want to compare this energy with some experimental results, with some experimental data. And when we compare this energy with experiment, we always have to keep in mind that this energy in experiment, this is free energy, thermodynamic free energy, which includes also entropy effects and so on. And the energy that we calculate in molecular docking is something different. And can you say a few words about this discrepancy? And probably you have some method, or you have some experience with comparison when we can do this, or when we cannot do this comparison. What you can say about this? Thank you.
On my opinion, there are no sense to compare the experimental and data from molecular docking. Molecular docking is a method that can compare the results within these methods, within this core function. We get the values of the molecular docking scores in the value near probably near approximate 10 minus 10 kilocalorie nmol, or is the available, and it's a normal value of energetic contribution, but their absolutely value is not any sense. It's only in comparison.
I see, I see, I see. So
We have many discussion last time when we now can do only molecular docking. It's very hard to us do molecular dynamic simulation for different our situation, but molecular docking is very valuable for us, and it's very valuable for their results, but they haven't any sense in the opinion of the real energy.
yes, yes, yes. I completely agree with you. And actually, but I have some. I read some papers, some literature where such attempts were done, and always I have as a question. So how it may be?
It's look like the attempt to do more precise the calculation of free bending energy in molecular dynamic simulation. It also a very large issues with these parameters, and we see the continual process to improve this possibility. It's as I can say, you have trials and reals, and only then we can get a successful result. It's not the last result, it's the result for improving and for
yes, yes. I understand. I understand. Thank you. Thank you very much. Thank you very much.
Okay, so thank you, Sergey. Are there any questions from the Kiev?
Probably somebody, Bogdana, you wanted to ask maybe. I have a question, but maybe after people who are
maybe in chat.
Maybe in chat.
In written form is more.
Okay, I have also one question, or maybe two. So you mentioned in the investigation of DNA lead that you use also Monte Carlo simulations and combined with molecular dynamic simulations. So could you please tell us which Monte Carlo method did you use? Are you use this Metropolis Monte Carlo or configuration?
Monte Carlo is a method which I learned from Danilo Victor Ivanovich about holes. Sergei Nikolaevich told yesterday. It's a very old program and our own software with our special potential function. So it's not development now. It's an old result, and molecular dynamics, we used the new program and new possibilities from Amber, from their working groups. Every year we have a new versions and new possibilities to use their new potential functions and new parameters for atom types of atom. But Monte Carlo is very old, and from 2018 I don't use it at all.
Okay, thank you. And the second question, you always combine molecular docking with molecular dynamic simulations, but you perform first docking and then molecular dynamic simulation. Did you try to do to first perform molecular dynamics and then molecular docking to see the difference in the configuration?
The molecular docking always for us is the start point. We do molecular docking and then choose some interesting structures. They are the start points for molecular dynamics. It's our procedure as a rule.
Yeah, I just think maybe sometimes it's necessary for some flexible proteins to do molecular dynamics.
Yes, it's very difficult to do flexible docking. A ligand flexible is a normal procedure, but a flexible protein is very difficult. So during molecular dynamics, we can do all our molecular dynamics only with the DNA. The molecular dynamics with protein, only one example when we modeling the complex with RBD domain and binding sites of RBD with SA2 receptor. It's only one experience, only one publication of this type.
Okay, thank you.
So
And
our experience is very, very small.
Okay, thank you very much. And we have also one question from the Zoom. Francesca, please. You can ask. We don't hear you, Francesca. We don't hear you. Please unmute yourself. Thank you.
One sec.
Francesca, we can hear you. No,
Okay.
yeah.
Thank you. Can you hear me now?
Yes,
Yes.
now we can hear you. Yes.
First of all, I want to thank you, Anna, for this very interesting talk with this impressive and fascinating journey through also the history of drugs. And I enjoyed it very much. So thank you. And the second is a comment, a kind the request of comment from you about the docking. About the docking, when we perform docking in my group, sometimes on the molecule that we study, we start from a crystal structure. But we know that in the solution, some portion of our molecule, our target is DNA in general, but not necessarily. Some portion in solution might have different conformation. So we perform the molecular dynamics, and then we perform the docking on several structures from the molecular dynamics. What is your experience about this? Can you comment on this procedure? Do you also perform in this way?
We try to do such procedures, docking, dynamics docking, and we don't publicate now our result for difference. It's not satisfactory us. No, it's very difficult for many reasons, and we haven't any experience. For docking, we even write a part in a monography, but it's only docking, in very detail DNA and different types of drugs or ligands with different modes of binding for intercalation and mineral bindings, but very small experience to bring it to the molecular dynamics. And reverse from molecular dynamics to Dawkins, we only some probes, but not results.
Yeah, I don't know, because in our case, I know that the drug might enter in some position that in the crystal structure are, for crystallization reason, maybe closed. So it's
Yes. As a rule, each molecular docking gave us some sets of structure with the equal energy scores, but very different structure. Yes, we try to do molecular dynamics for different structures. But then in sometimes they goes to the one structure, to very similar structure during their feeds to each other. But
Doing the
they're very
dynamics.
small experience and not publication result.
Thank you very much.